All right. Good morning to everyone here at the Everglades Research Center and folks who have connected online as well. On behalf of the seminar committee here at the Everglades Research Center, we are trying something different for the next 10 series of seminar or so, where we are having faculty and extension agents of EREC present programmatic seminars so that we all can learn and integrate our science together. So to kick things off, we've decided to go first. And today it is my pleasure to talk about integrated soil, water, and nutrient management as pillars of sustainable agriculture. My name is Jehangir Bhadha. I'm an associate faculty in the Soil, Water, and Ecosystem Sciences Department. And one thing I want to add that as we go through this presentation, I'll have some of my students and postdocs jump in for a couple of minutes to highlight some of the work that they are doing. Okay, so I think we strongly believe that the future of sustainable ag depends on three vital components: soil sustainability and soil health, very critical; water resource and water quality, just as important; and nutrient management. I think it'll take efforts in all three of these areas for us to promote sustainable ag in the future. And why is soil so important? Let's focus on soil first. Why is that so important? Because annually, USDA has shown that almost 24 billion tons of topsoil is lost globally. And there are different ways in which this can be lost. One of the main reasons is wind erosion. You can have runoff through storms and hurricane. And then local to us here in South Florida is this effect of soil subsidence related to our histosols. I mentioned histosols. Most of y'all here in the room obviously are aware of it and work with them practically every day. But these are organic soils. They contain upwards of 70, even 80% of organic matter. Underneath this organic soil is the bedrock limestone, which is calcium carbonate based. Hence, the pH of the soils are fairly high, neutral and even above that. So if you're talking about soil pH in this region, you're looking at 7.5, even pushing 8. These soils were drained back in the early 1900s to accommodate for farming in the region. And as a result of that, it triggered the process that we locally refer to as soil subsidence. Why is soil health and sustainability so important here? Well, there is a whole host of reasons that we've listed here, and there could be more: reduced fertilizer inputs, less weed pressure, moisture retention, soil stability. But if we look at the bottom two, personally, I feel those are very, very critical, regardless of what soil type you deal with. That is increased soil organic carbon and increased organic matter. It's important because carbon is the centerpiece or the center stone or the key that binds all the other elements, all the other vital nutrients that the plants need. So the more carbon, the more organic content your soil has, the better chances these elements, these nutrients can stay sequestered in the system. So what we've started to do in our program is try to evaluate some land management practices that are beneficial for improving soil health. This could be in the form of cover crops. This could be in the form of application of amendments, crop rotation. Whatever the practice is regionally, we can step in and evaluate the practice to see if we are seeing benefits or maybe some drawbacks associated to those benefits. So we have a treatment setup, we have a control setup, we do pre-post soil sampling, we analyze different soil health parameters that have been outlined by the Soil Health Institute. We generate that information. Obviously, we publish it into scientific journals, but we also make sure we deliver that information to our stakeholders as outreach. Some of the parameters that the lab is efficient in conducting have been listed here. Most of them, as you would see on this list, are either physical or chemical assessments. Only recently have we started to work on some biological assays, especially enzymatic assays from a soil health perspective. And some of the students will talk a lot more about that. So one thing to keep in mind is all of these indicators are conducted in-house here at the research facility. So again, for folks in the audience or in the call, if that's something you guys are interested or want to do, feel free to reach out. About 8 years ago, we received some seed funds through the Southern SARE program to evaluate soil health effect related to cover crops. And so we got in touch with a whole host of growers across the state of Florida. We invited extension agents to help us facilitate some of the soil sampling work. And what I wanted to basically highlight are the two pictures that you see at the bottom here. There is the same grower that has left a patch that is fallow. You can see the sandy soil in Loxahatchee. And then right next to it, he's grown a mix of cowpea and sun hemp. And this is probably not at mature, but at mature, this cover crop can go as high as 10 feet. So clearly, just visually, you can see the practice of cover crop can have as far as land management is concerned. And what we found as far as results, and we don't want to go too much into the qualitative aspect, but from a just by looking at these numbers, we found that we were able to evaluate these indicators through the different collaborative growers. 6 out of 9 farmers showed that there was an increase in our malic-3 phosphorus. 4 out of 9 showed increase in cation exchange capacity. But at the same time, only 1 farmer showed that, hey, your potassium was going up. So there are some benefits, there are some losses, and we want to make sure we kind of take all of those into consideration. I'll hand it over to postdoc Dr. Tanjila to kind of give a little snapshot of current projects going on as well.
Thank you, Django. Hello, everyone. I'm Dr. Tanzila Jasmine. I'm a postdoc in Dr. Bhadha's lab. So I have been involving with soil health research, which includes cover crop and its impact on soil health in Florida. So we are conducting a 2-year study, which is funded by Florida Department of Agriculture and Consumer Sciences. This is a study where our goal is to assess the soil health under cover cropping during the fallow periods. To conduct this study, we have been collaborating with other research and education centers as well as county agents. Our collaborators are supporting our project by identifying the growers, supplying soil samples, and also developing the project goals. So to represent the Florida soil, we are collecting samples from both northern and southern part of the states. To assess the soil health, we are analyzing soil properties based on the biological, physical, and chemical indicators of the soil. All these health indicators, after analysis, we are using different techniques or models to quantify the soil health indices. Recently, we have submitted a manuscript in a scientific journal where our major findings were we observed that active carbon in mineral soil was lowered by the short-term cover cropping, whereas we observed significant increase in water holding capacity of the mineral soil. So currently, I am more interested to dig a little bit down to understand how cover cropping is impacting the soil microbial community structure and the enzymatical activities, which are more majorly involved in the major nutrient cycling in the soil. Beside these scientific experiments, we are also collecting farmer survey to understand their feedbacks and also encourage them to continue cover cropping during the fallow periods. Thank you.
Thank you. Yeah. Thank you, Dr. Tanzila. Okay, let's shift gears to another interesting topic that we've worked with our stakeholders here in the Glades is bagasse. Bagasse is a byproduct of sugarcane production. It's a cellulosic material, and a lot of it is generated here in the region. So about 5 years ago, we had a conversation with US Sugar, and we wanted to test the effect of bagasse from a soil health point of view. Initially, what we did was we just looked at the bagasse itself for its chemical properties. What does it contain? And you'd be surprised, it does have significant amount of nitrogen, phosphorus, and some other microelements present in it. Here are some images of how commercially growers are applying this back in the field. The pile of bagasse that is generated each year, more than 2 million metric tons of bagasse is generated annually. So that's a lot of bioproduct that we can kind of put back and reuse in the field. Looking at preliminary results, I'd be the first to let you all know that it is not all positive when you add so much of organic matter back into the soil, because you're putting this carbon back into the field. There is potential for your nutrients to once again be bound onto this carbon, onto this organic matter. And therefore, you can treat bagasse as a slow release rather than a one-time instantaneous amendment. Looking at the Everglades agricultural area, again, for the audience present here, you all know this like the back of your palm. But for those that have connected online, the EAA comprises of almost 450,000 acres of organic soil. 80% of that is used for sugarcane. Our topography is very flat. There is the limestone bedrock underneath. These soils were actively drained for farming, and the water leaving the EAA is routed through the STAs. Those are the stormwater treatment areas shown here in dark green before it is delivered to the water conservation areas that are these large water bodies in blue that you'll see in the picture. Irrigation is unique in this area. Why is it unique? Irrigation and drainage, because it's bottom top. Unlike the Midwest or the rest of US, where you see irrigation lines overhead feeding farm fields, this is all driven by gravity from bottom to top. So if a grower wanted to bring water into the farm and irrigate his farmlands, he would turn on the farm pumps. Water would flood the main canals, which would then get channelized into the ditches, and those ditches would then seep the water up into the farm fields. Inversely, if you wanted to drain the farms, you would turn the pumps, discharge the water off-site. That'll lower the water in the main canal. The ditches will start draining the fields, and your fields would have lower water table. All of this water is kind of looked and monitored by South Florida Water Management District. And farming in the EAA is also very unique. And this image kind of summarizes that. It's not just a monoculture. I do mention sugarcane is the primary crop, but it's not just the monoculture of cane here in the region. We do a lot of rotations as well. And to kind of bring that all home, what we are showing here, if you're looking at a 4-year cycle, let's say in October, you planted the first sugarcane. That cane we would call plant cane for about 12 to 14 months. Once you harvested that, you would have another crop of sugarcane growing from it. That would be your first ratoon. Another 12 months pass by, and another crop would emerge from the same plant, and you would call that your second ratoon. Depending when that second ratoon gets harvested, there is potential for that same plot of land to either go in as a winter vegetable. There is a whole host of vegetables that we grow here: sweet corn, lettuce, green beans, radishes. And if it is around the summertime, April, May, June, July, there is potential to grow flooded rice in the region. So again, I brought up this topic of flooded rice, and I mentioned cane. So there is some synergy going on there as well. Lots of benefits of growing flooded rice in the region, and decades of work has gone into that to evaluate that. It was 1970s, late 1970s, when rice was first introduced here in the EAA as a rotational crop. And it is definitely a good crop to grow in rotation with sugarcane, because almost 50,000 acres of land is fallow potentially here in the summer months. Why is it fallow? Because it's just too hot. You really can't grow any winter vegetables here in our hot summers. So there is potential to come in, flood these fields, and instead of leaving them as flooded fallows, growers can actually cultivate flooded rice in the region. An interesting point about flooded rice is here, grown, cultivated here in the region, is listed right there at the bottom. No N, P, or K is applied to grow rice in the EAA as starter fertilizer. So where does rice get all its nutrients from? Well, it either gets some of the legacy nutrients that the prior crop has left behind. It gets the nutrients from the soils once they are flooded, or it gets the nutrients from the irrigation water. So that kind of brought us into evaluating a long-term study, conducting a long-term study to evaluate conservation efforts related to rotation of sugarcane and flooded rice. This is an NRCS-funded project. And again, we have collaborators, Dr. Jehangir and Sam Smith, that are working on this. And with that, we have a graduate student, Shua Bai, who is also involved in this project. And the project objectives are to coordinate plans with our stakeholders, establish monitoring activities, facilitate the conservation practices, record these practices, and then evaluate that information. With that, Shua, I'm going to hand it to you. If you can go through the next couple of slides. There you go. All right. Yeah. You think
hello, everyone. My name is Bai. And my PhD research, as you see in this figure, we work on the sugarcane and the rice rotation. Here, we installed the monitoring wells in the sugarcane field, and we can monitoring the water levels from those fields. So we can know the transportation of the nutrient and the water from the canal and between the canal and the field. And you can see in the left map, here has some points which shows, which shows our monitoring locations in 5 fields from the EREC. And the middle one is the monitoring wells. We installed the monitoring wells in the field. As you can see, there are some filters on the pipe, so the water can come in, but we can prevent the soils comes in. So my research, my PhD research focused on the assessment of sugarcane and flooded rice crop rotation effect on soil and water quality. We know that in the Everglades agriculture area, sugarcane accounts for 80% of the cultivation, while soil loss due to oxidation is still a great concern for the future farming. How does the sugarcane cultivation impact the nutrient mineralization? Does the soil loss can affect the crop growth? And we know sugarcane and rice rotation can slow down the soil loss rate. But what is the practice effect on soil quality and water quality? Those 4 questions need to be answered in my study. Many people has asked me about the second question. Can soil depth affect crop growth? To answer this, we collected the NDVI from the satellite alongside the soil depth measurement from 5 fields in EREC. And our findings indicated that there's no significant difference in crop growth between shallow and deep soil. This is a surprising result because we usually think the more soil can provide more nutrient and water to the crop. But one day when I asked the same question to a farmer and he gives me the same answers, he said, soil changes, but we also adapted. It's clearly that the modern agriculture practice in, for example, the soil testing program, the plant breeding, and effective water management have overcome the challenges posed by shallow soil. But if the soil one day is depleted, that's another story. So if you want to get more information about my research, feel free to contact me. Thank you.
Thank you. You can hold. All right, we'll shift gears again and we'll talk a little bit about phosphorus. So welcome to the STEPS Center. I want to go through the next few slides, talking a little bit about this project. This is an NSF-funded project that involves 9 institutions across the nation. University of Florida is one of them. And University of Florida also plays a key role in this project because it is one of 3 TBL sites. TBL acronym standing for triple bottom line. Especially South Florida, where we are working in phosphorus, is very, very unique and essential for this center's success. So STEPS is a convergence research center with phosphorus sustainability as its vehicle. What this means is that we are approaching the topic of phosphorus sustainability from different angles. We are not just working with soil scientists or water scientists, but we are also bringing in human health folks, social scientists, engineers, data and informatics folks to come up with solutions and answers. We all know that all living beings, plants, humans, animals, we all need phosphorus as an essential element to live. However, this use of phosphorus is unsustainable, and if used in excess, can be harmful to the environment. We know that phosphorus is mined as a non-renewable resource. It is essential for plant growth, but only about 20% of that phosphorus ultimately makes it into the human food chain. So 80% of it is lost. And where does it go? Either stays in the soils, in the systems, or runs off into freshwater systems. 50 years ago, we were mining about 2 million tons of phosphorus per year. And today we are almost 20 million tons of phosphorus per year. So the demand is high. Our approach of handling phosphorus is single-use extractive process. So we mine it, we use it, and we lose it. Interventions need to be in place for us to recapture this phosphorus and be able to reuse it. So there are many ways in which we as scientists can step in and help with phosphorus sustainability. The goal or the vision of STEPS Center is 25 in 25, which basically states that it will facilitate a 25% reduction in human dependence on phosphorus and a 25% reduction in losses from point and non-point sources over a period of 25 years. It's a challenging goal, but I think the team of folks are ready for it. The way we are approaching this challenge is looking at 17 orders of scale length. So there are teams that are working on nanoscale related to phosphorus capture and reuse, and there are teams that are working on a global scale at the systems level to understand how phosphorus is flowing at a global level. Again, when I mention team, it is clearly a team effort. A lot of researchers, a lot of scholars, students, postdocs are involved in this mission. There is a whole host of projects that have been funded through STEPS, and I don't want to go through the list of all the different projects listed here, but I just want to point out one, project number 2.5, which is controlling and utilizing legacy phosphorus in soils. And that one could argue that a lot of work has been done even historically by our Soil and Water Sciences Department, and now that some of us here at the Research Center are conducting. So with that, I'll hand it over to Anik Mahmood, who is a PhD student, STEPS funded. And Anik, you can tell us a little bit about your project.
Good afternoon, everyone. Welcome to the presentation. And today you guys will know, apart from going to the South Florida beach, what I am doing in the research center. So the project, the PhD project title of my is that the unlocking the chemistry of legacy phosphorus to maintain sustainable agriculture and healthy environment. Here you can see in the slide, in the right side, where what is the importance of the phosphorus. Phosphorus is an essential nutrient which is necessary for photosynthesis, respiration, reproduction, cell division, and plant root development. And worldwide, there is about 71 million metric tons of phosphorus reserve, but where USA has only 2% of it. And it has estimated that this current reserve can be depleted in near about 100 years. And that make a legacy phosphorus is very promising resource to unlock, because this you can see that more than 70% of the land, our arable land, are phosphorus deficiency, and globally applied phosphorus fertilizer is about 17 million metric tons, and only 10 to 36% of that used by the crops, and rest of them are fixed into the soils in an estimated rate of 10 million metric tons. And from there, I have 3 chapters that I want to do related to the phosphorus. Here, the first chapter related to the chemistry of legacy phosphorus. The second one related to the release of inorganic, organic phosphorus. And the third chapter that I want to do is related to phosphorus capture. Here, now I am showing you some of the results that I have generated so far. In the x-axis, you can see the concentration of P in milligram per kg, and the y-axis is the different sample place that I have. In the A, as you can see in the figure in the A, it's related to the acidic soil of North Carolina, and the B and C is the soil we have in South Florida, the muck soil, and the D is calcareous soil of Arizona. Just in one look, you can see that in calcareous soil of Arizona, we have most of the phosphorus is calcium, magnesium, phosphorus in the red mark. In our soil, in the muck soil, we have most of the phosphorus fraction is residual P. We marked here in the black color. And on the other hand, in the acidic soil of NC, we have humic and fulvic P. With the facility in the Ambris facility in Gainesville, we have done some advanced chemistry work using solution NMR. Here you can see the South Florida soil sample that where we use. We have 2 depths, 0 to 15 centimeter and 15 to 30 centimeter. Here you can see we have a lot of organic phosphorus present in South Florida. So still we are very early in the stage, but we are still learning more about the chemistry of legacy phosphorus. So thank you so much.
Thank you. Okay. All right. Let's highlight some more collaborations, programmatic collaborations. Here is one that we have started to work with Dr. Zach Brim and his team out of TREC, looking at hemp nutrient management, development, and guidelines for the state of Florida. Again, it's a collaborative effort. Dr. Hardeep Singh, Lakesh Sharma are all involved in this. Hemp fertilizer guidelines, there is quite a bit of information related to nitrogen, related to phosphorus, related to potassium. But there are caveats associated with this. These studies, these research was conducted out of the state of Florida historically. And so for us to kind of evaluate this here in the state, it is critical to conduct these studies here in state. And most of them were conducted for grain and fiber. But here we are also looking at other products like oil as well for hemp production. Some of the preliminary work that has been conducted across 3 sites here in Florida has shown that about 100 to 150 pounds of nitrogen per acre seems like a good sweet spot for hemp to grow. Again, these are preliminary work that has gone on, but clearly hemp requires nitrogen as a nutrient to grow. From an outdoor experiment on grain and fiber, once again, numbers seem to fall and show us that about between 100 to 150 pounds of nitrogen is necessary for hemp to grow successfully here in Florida. So what are some take-home messages? Well, preliminary research does support that about less than 150 pounds, you don't want to go more than 150 pound per acre rate. Split application is a good practice. You don't want to apply all of that in one go. More does not always mean that it's better. And also watch out for micronutrients. There are a lot of micronutrient deficiencies in hemp that have been identified, and further work needs to go into that. Another group of work that we are conducting is with folks in ag bioengineering, Dr. Zivan Yu, Sandra Gruzman, and the team. And we are looking at how to evaluate carbon sequestration, how to address the topic of carbon credits and management out in the field. Obviously, funding is essential to take on this challenging topic. So we've put in grants with NSF to see if we can conduct this project on a long term. There are two main factors that will be very critical in the success of this project. One is the tech factor, right? What is the technology out there that we can leverage to address this topic of carbon sequestration and carbon farming? But then the second aspect is the social aspect. How welcoming or how open are the growers, are our stakeholders, is the public in wanting to learn and wanting to address this topic on carbon sequestration? So with that, we have UFIFAS extension, about 4 years or so, is trying to spearhead this team, a group of extension agents and specialists on the topic of climate and carbon extension educators, or C2E2. So again, it's this team of folks that are trying to set the stage, provide that preliminary information that our extension agents can take to the stakeholders and say, hold on, if you all are interested to work on the topic of carbon capture and carbon credits, then this is how we can go about doing it. There are some aims associated with it. Obviously, what are some ways that we can actually capture and account for this carbon stock, making it aware for our public, our growers, and the data rights issues associated with it? How do we incorporate carbon farming in the normal, typical farming practice of cash crop? And then providing that educational component associated with it. Again, this is no strange slide to folks that have been working in this. When you're talking about carbon capture, you're going to have to deal with it above ground, but also there is a component that deals with what's happening below ground as well. You can look at it from an on-farm data assessment. You can do some ground truthing of collecting samples and bringing them back into the lab and testing them. And you can also use some modeling tools and AI that can give you some answers. All three approaches are currently in place and actively being researched. So with that, I'm going to hand it over to graduate student Suraj, and he'll tell you a little bit more about what he plans to do as part of his PhD.
Thank you, Dr. Django, for providing the opportunity. Myself, Suraj, a second-year PhD student under Dr. Django's lab. And my PhD study basically about determining the soil organic carbon dynamics in the Everglades agriculture area using the data-driven machine learning modeling and the spectroscopy. So basically, as Dr. Django was telling about the problem of soil subsidence and how we are losing these soils. This is one of the basic problem of EAA. And this has been reduced, as you can see in the left, it shows the subsidence over time, and it has been reduced, but it is still a concern, and we are still losing the soil. So to deal with this loss of soil and to understand the subsidence, we need to understand the soil organic carbon, and we need to understand how stable it is. So this loss of soil in EA brings up with the following research question. First of all is what is the proportion of the stable carbon in EA? The second question is, what are the specific mechanisms which are stabilizing this soil organic carbon? And there are basically two mechanisms as shown in the picture on the right, chemical protection and physical protection. We also need to understand what are the relationship between these protection mechanisms. To solve these research questions, we have the following workflow objectives, how we're gonna deal with this project. First, we will try to investigate the carbon stability patterns in EA using the soil organic carbon fractionation. Then we will scale up the data for whole EA using the spectroscopy. In the next step, we will use data-driven machine learning approaches to further scale up these patterns. And finally, we will use these models we developed to compare the different land management practices in EA and how it is affecting the soil subsidence. A more comprehensive picture of the whole project is shown in here. As you can see on the left, we will have samples from different locations. We will go through the lab analysis as well as we will collect the spectral data. We'll use both of these data together and we will term them as measured data. On the data, we will also try to access the available geodatabases, which will be available online. And we will try to combine these measured data and available geodatabases using data-driven AI models. These models are basically regression models, and these models will try to relate these data together and help us get the soil organic carbon map for the current for now. We can also go for the future projection, where we can use the projected climate data, land use data, and other variables, and the current developed model we developed, and we will get the maps for future, as you can see there. We are also planning to use simulation modeling tools like DSET in our project, and we are hoping for getting some good results. And if you have any questions about this research, you can contact me. And now I will hand this to Noel. I think he will be presenting the research.
Good morning. Good morning, everyone. My name is Noel, a PhD candidate under Dr. Bhadha's PhD program. Before I get started, I would like to acknowledge 1,000 SAR for funding my PhD project. This PhD project was conceptualized based on the one research question. Can we slow down soil subsidence? In order to answer this question, we do need to come up with two potential solutions. One is to enhance carbon input through carbon sequestration, and also to decrease carbon output through suppressing microbial activities associated with organic carbon mineralization. So my project is all about trying to understand how different cropping systems we do have here in EAA are impacting carbon input and also microbial activities associated with carbon mineralization. So as you can see, these are on the left side, these are different cropping systems we do have here in EAA. And we collected pre and post samples, trying to understand how they are impacting microbial activities. So we measured the 4 enzymes, and you can ask yourself, why enzymes? Just imagine yourself in the restaurant sitting in front of a big chunk of meat. What do? You get a knife, right? So same goes for the enzymes. So microorganisms, they need carbon as a source of energy. But this carbon is too big in the soils, so they can't take it into their cells. They don't need to produce the enzymes, which helps them to break down these large size carbon into a small piece so that it can be taken into their cells and then they get energy. Once the carbon is uptaken into their cells, it is oxidized into CO2, thereby getting volatilized into atmosphere. So as you can see, the first 2 enzymes, Glucosidase, it involves in breaking down cellulose into glucose. Glucose is source of energy for most of soil microorganisms. The second one, glucosaminidase, it breaks down chitin into amino sugars. Nitrogen is very important for microbial productions, and also the sugars they use for microbial activities. The last two, phosphatase, phosphominization. The last one, sulfatase, is into sulfaminization. As you can see, the red cycles, it shows that flooded rice significantly decreased most of microbial activities. On the other hand, as you can see, the sugarcane significantly, apart from glucosidase, significantly increased the rest of microbial activities. For now, we don't have any conclusion we can draw because we cannot rely on one single source. We are trying to replicate the study. Once we get the second year data set, then we can come up with some conclusion to see if these cropping systems, they are sustaining our agriculture production for not only for us, but also for the future generations. Thank you so much.
Thank you. Okay. We had the opportunity to work on the DeLuca Preserve. Again, a collaborative effort with Dr. Anna Braswell, Dr. Will Mertens, and Sam Smith. This was in its very first year when UFIFAS Research Office provided some seed funding for us to go in there and conduct some work. 27,000 acres of land in South Central Florida, you can say, right at the corner of 60 and Turnpike, Eha Junction for some of us that commute up and down to Gainesville. And so that is demarked here in yellow, the DeLuca Preserve. It is really, really beautiful preserve there. There are so many different types of land covers and land practices that from a soil scientist point of view, it's just wonderful to go in there and get all this diverse soil to be able to conduct some work. Shown here geospatially are the different land covers of the different cropping types that are present there. And so we went in there and did some preliminary work associated with this project. Our goal was to evaluate the baseline soil health characteristics of the different landscape. Dr. Mertens and his team was looking at microbial activity associated with that diversity. Dr. Braswell and her team was looking at the hydrology and how the wetlands are expanding and contracting and that effect on the biodiversity. And Dr. Schmidt's task was to take all this information and produce it geospatially and also create like a data repository. Sampling work was conducted on 2 seasonal periods, a dry period and a wet period. And some of the landscape, like I said, was extremely diverse. These were some of the different landscapes across the 27,000 acres that we collected soils data from. Again, today is probably not the day to jump into the data, but clearly we have started to evaluate some of the soil health parameters. And based on the different land practices, we have started to rank it and then relate that with some of the soil microbial work that Dr. Mertens and his team is conducting. Like I said, hydrology is important, and Dr. Braswell's group is working on seeing if there are some ecosystem services that can be provided across the different landscape of the DeLuca project. And then Dr. Sam Smith and his team are going to kind of accumulate all this information and generate that very baseline repository for the DeLuca Preserve. I am aware that subsequent years of funding have been offered through IFAS at this preserve. So it's a really wonderful site to be there. Okay, so that group is also growing. Again, you can see it's an integrated work that the program has been involved with. I want to take a few minutes to talk about the lab and some of the work that we conduct in our lab as a programmatic effort. Dr. Abul Rabbani, who couldn't be present here today, but he's the chemist who oversees pretty much every single sample that comes through our lab. Some of the instruments that we have in-house to work on is the ICP OES. We have OPS personnel, Johnny Rumpf, help us with some assessment. We have a discrete analyzer that can evaluate all these different parameters, and I see a whole host of students here in the group that have used this instrument for their data assessment. Savannah Tora is another OPS student, and she's also a DE master student that spends some time in our lab conducting work, and she does a lot of work using the Shimatsu analyzer, which is an instrument that looks at the liquid phase for carbon and nitrogen assessment. Some benchtop parameters, and then just the new team member on our program is Livio. Livio is our field technician, really helpful when we conduct field work in the field. And there's Dr. Rabbani working on a dry combustion carbon nitrogen analyzer as well. So again, I wanted to highlight that teamwork of our program. And then there is extension. All this information that we generate, we make sure we hand out this information to our stakeholders, all forms, all ages. How do we go about doing that? Obviously, we do some needs assessment to begin with, some surveys to begin with. Once we gather that information, we conduct the research, and then we hand over the results through fieldworks, one-on-one trainings, in-service trainings, etc. A few years back, we conducted an extension in-service training. It's training the trainers, so to speak. We train our extension agents, getting them ready so that they can go out and talk about soil health and sustainability with their farmers and with their end users. Last year, 2023, we did a similar training on the topic of science and technologies for phosphorus sustainability. Again, that was well received on campus, and we found almost 23% increase in knowledge change from the folks that attended that IST. On lines of extension, I've been involved with the ESP group, the largest extension development community in the country, Epsilon Sigma Phi, and a lot of work that has been done internationally related to extension. So if folks in the room or on this call are interested to be involved with extension activities at an international level, please feel free to reach out, and I'll be happy to direct you all in the right direction. So it's the oldest and the largest organization for extension professionals. And there is a whole host of committees. I've been involved with the bottom one, the Global Relations Committee, now for the last 5 years, and a lot of exciting work has been conducted and is in the pipeline with ESP. With that, I'm going to end. Once again, I want to acknowledge that this is a programmatic effort. For the next few months, we are going to see faculty of EREC present programmatic theme-based research work. And I also have a whole host of folks that I would like to acknowledge who at every stage have funded our research and also our outreach efforts. Thank you very much, and I'd be happy to take any questions. Yes, sir.
All right, my name is Dennison. Thank you so much for the great presentation. I wanted to ask a question on the soil subsidence issue. I understand that you are currently monitoring the soil subsidence using the subsidence post, for example, that is right there. And I understand that the subsidence post normally monitors the subsidence of the regolith, which is the part of the soil above the bedrock. Now, there's also type of subsidence that happens due to the overdraft of the aquifers, like bedrock itself, the space within the bedrock itself. If that space has a lot of water, that means subsidence can happen. So is there anything that is happening that is allowing us to monitor the subsidence caused by this overdraft of the aquifers?
Yeah, you know, I did my master's in geology, and one of the topics that we had learned as far as Florida geology was the karst topography. Karst topography is basically what you are referring to. We've got this limestone bedrock underneath, and because of that karst topography, it is very common that certain areas might see depressions, where you can have depressions because of the bedrock being subsided, and then all the soil with it gets lowered and subsided with it. That does happen. Sinkholes have opened up, as a matter of fact, as well. Whether we are monitoring that here in the EAA, whether our program is doing it, no. We are basically looking at the subsidence that is above that limestone, above that lime rock. I also want to add that this rate of subsidence is not uniform. It has changed. Even if you look at that subsidence post right outside of the dormitory, it states that historically this subsidence was a lot more accelerated, a lot more faster. And over the years now, after 100 years, that rate of subsidence, so to speak, has slowed down. And that's because the soil quality, the texture of the soil has changed. That's one of the reasons, right? Another reason is that the soil itself has been compacted. So now there is more material in the same volume. So all those factors play into, answering that rate of subsidence that you're talking about. But yeah, there are two things going on. One is like a geological subsidence, where you've got the bedrock karst system that can subside. And then you've got this soil loss via oxidation, which we locally term as soil subsidence as well. Thank you. Yes.
Thank you, Django, for a very comprehensive program overview. I actually have a question for Tangela, if that's okay. Yes. So you said active carbon decreased with increased cover crops. So can you talk a little bit about if this is a good thing, a bad thing? What does it indicate in terms of the impact?
Yeah, that's a very
And what is
good
active
question.
carbon? Maybe some people don't realize what active carbon is or what does it indicate.
So the active carbon is the bioavailable form of the carbon, which is most which is vulnerable to loss, I believe. So as it lowers in the mineral soil, so that may have like help to reduce the carbon loss in mineral soil. So the cover cropping actually that we observed from the previous 2-year study that is significantly lower down the active carbon content in the soil, and which may help to maintain the sustainability of the soil. Yeah, that's a good thing. Yeah.
Yes, go ahead.
First, I would like to congratulate you for such a great presentation and for having, to me, a fascinating research program. I have several questions, but I think my major question is about soil subsidence. I know there are several factors that contribute to subsidence. Do you have data, or can you speculate what is the major drive of soil subsidence here, and what proportion could you attribute to microbial activity?
Sure. So soil subsidence as a definition, as a simple definition, is soil loss, right? If that soil loss from 9-foot concrete post, now we are at 6-foot exposed, so we've lost 6 feet. So it's soil loss. Where has it gone? Some of it during the hot, dry period is lost by wind. Wind erosion can blow off soil as well, and you can have soil loss. Even that would be accounted as soil loss. It could be just the one event. You can have a hurricane, and that hurricane can wash some of the soil from the fields into the ditches and canals, and you can have loss associated to that as well. So those are fast processes where you can have soil loss. But I think some of the most persistent, constant processes, like you mentioned, is the microbial process. It is the microbes that are slowly and steadily breaking down that carbon, and that carbon is getting respirated and ultimately mineralized back into the atmosphere. So that is kind of the main driver over the century.
So just have a follow-up question. Okay.
So in addition, there might be compaction and shrinkage of the organic matter too. So that adds the cell subsidence, originally. So there's shrinkage and compaction too.
Yeah, so when you talked about adding bagasse, wouldn't that kind of accelerate or feed the microbial activity?
Absolutely, absolutely. If you like, how were these soils formed, right? These soils were formed geologically over 17,000 years geologically, where this entire basin, this entire South Florida was a bowl, and organic matter accreted over the geologic period. If we add that much, realistically, it is impossible. It is just not possible to haul in hundreds of thousands of trucks of organic matter to recreate those soils. But wherever the opportunity lies to add amendments back into the field, I think we should take those on. Wherever the opportunity lies to plant cover crops and have living roots in the system, we need to jump in and take that opportunity, because fallow fields are the worst enemy of soil subsidence. Yes. Yeah, I was just going to ask if there were questions on Zoom. Anybody on Zoom, if there are questions or want to speak up? Okay. Okay. Yeah, I think we're good. All right. Thank you all very much, and enjoy the pizzas outside.